Cancer and Oncoven by Rae'Miah James. What is cancer? Cancer is a disease which is in the body. The body cells grow uncontrollably and spread to other parts of the body. Normal human cells grow, which multiplies, and following the cell division, they will become old or damaged and die off, following the new cells. When cancer begins, it is usually when there is an abnormality in the cell division, and apoptosis, which is programmed cell death, does not correctly get rid of cells. So many cells may form tumors, which can be cancerous or not. The cancer spreads and invade and metastasize, which means moving to different places in the body, forming new tumors inside of the body. Cancer affects 1 in 3 people in the United States, and you have 2 types of cancers: blood cancers such as leukemia, lymphoma, and multiple myeloma. You also have solid tumor cancers with other body organs or tissues, such as breast, prostate, lung, and colorectal cancers as well. What is the CHOP method? The CHOP method is the combination of medications for chemotherapy drugs that uses cyclophosphamide, which is the C, doxorubicin hydrochloride, aka hydroxydialmycin, which is the H, vincristine sulfate, which is Oncoven, which is for the O, and the steroid hormone prednisone, which is the P for CHOP method. Now, CHOP is used to treat non-Hodgkin's lymphoma, and it could be used with other drugs or treatments or other types of cancers. Now, CHOP works by destroying quickly dividing cells, such as cancer cells. The CHO in CHOP of the medication drips into the bloodstream through an IV. The P is given through tablets with or after food. Medications that could be given through the central line, PICC line, and portocath. Oncoven is also a drug that stops or slows down the growth of cancer cells by stopping the cells from dividing and making new cells. This drug is known as a vinca alkaloid. And there is the picture right there of just showing the CHOP working. Now, here you go. You have I just put in just the cell cycle. It was dealing with mitosis, since Oncoven does mess with mitosis and in some of the phases of the cell cycle. So you have interphase and mitosis. And as you can see, the mitosis is the division of the nucleus in cytoplasm so that a single cell forms into 2 identical daughter cells. Now, cell division, which is used with tubulin. The tubulin mechanism used in cell division with the alpha and beta monomers of tubulin exist as isotopes, differing in their amino acid sequence encoded by different genes. The alpha and beta heterodimers polymerize into microtubules, which are indispensable for cell division and growth. The mutations of tubulin isotopes expressed in invasive tumors affect the binding of anti-cancer drugs, which contributes to the drug resistance. The alpha and beta tubulins are post-transitionally modified in the cell cycle or stress-driven signaling pathways, which also regulate microtubule dynamics through interactions with microtubule-associated proteins known as MAPs, and including with molecular motors. Tubulin also plays an important part in the nervous system, dealing with health and neurodegenerative diseases such as Parkinson's and Alzheimer's. And then you have the picture right here of how a tubulin heterodimer is formed. Now you have Oncoven mechanism. The Oncoven mechanism is a naturally occurring vinca alkaloid. Vinca alkaloids act as anti-microtubule agents that block mitosis by arresting cells in the metaphase of metaphase phase. These drugs act by preventing polymerization of tubulin, which forms microtubules, as well as including depolymerization of formed tubules. Vinca alkaloids are cell cycle phase specific for M phase and S phase of mitosis. Now, it could be traced back to the 1950s, and then oncovin was approved by the U.S. Food and Drug Administration in July of 1963. Oncovin mechanism disrupts cancer cell growth and cell division. Another mechanism of action for oncovin includes when it interferes with nucleic acid and protein synthesis by blocking glutamic acid utilization. Oncovin deals with the micro. Now here is how my oncovin deals with the mechanism as well. The microtubule binding, which binds to the plus enzyme microtubules composed of tubulin subunits. The inhibition of microtubule polymerization interferes with binding of tubulin subunits within the microtubule lattice, prevents the addition of new tubulin subunits to growing microtubule, inhibiting elongation. The disruption of meiotic spindle formation during mitosis phase of cell division, forming meiotic spindle. It interferes with the microtubule, disrupts formation and the functionality of meiotic spindle. This cell cycle also arrests, which disrupts the spindle formation caused by oncovin. Treated cells cannot progress through the cell cycle. The cell cycle arrests in the metaphase stage. The chromosome fails to align and segregate correctly. You have the induction of apoptosis, which during the metaphase, it arrests triggers cellular response, leading to the cell death. This gives it an ability for the cell to complete mitosis during the microtubule disruption. Another is effect on rapidly dividing cells, which targets rapidly dividing cells depending on adequately functioning microtubules during cell division. So overall, this is how oncovin disrupts the cell cycle. Oncovin mechanism continued. You have when it deals with pharmacokinetics, which is dealing with the absorption, disruption, metabolism, and elimination. Now, with absorption, oncovin is given through the IV route and is absorbed into the systemic cell circulation. Disruption, the drug exhibits high binding affinity to plasma proteins, which is albumin, and blood cells, especially platelets. It has some limited penetration due to the blood-brain barrier and accumulates in the cerebrospinal fluid. The drug is distributed to various tissues, which includes liver, kidney, spleen, lung, and heart and muscles. The affinity, it has a strong affinity for tubulin, oncovin does, which it involves in cell division and transport. Now, the metabolism, vincristin, aka oncovin, undergoes extensive metabolism in the liver, primarily mediated by the cytochrome P40, also known as the CYP enzyme system, the CYP3A4 and the CYP3A5. Influence bodies, enzyme systems, as well as the drug interactions involving inhibitors or inducers of these enzymes. Elimination is eliminated primarily in the bile and feces as metabolites, with 20% excreted in the urine. It has a biphasic pattern with an initial rapid phase. The clearance rate of this varies among people and is influenced by many factors like age, weight, liver, kidney function, and other medications being taken. And this is just some pictures as well, as you can see with the meiotic arrest. And this is my citations, and